Good afternoon, all of you. So I welcome you all in the post-lunch session. Now we'll be having tutorials for 2 hours. Dr. Shukla and Dr. Howlu will, along with Dr. K.K. Sharma, will give some details about some experimentation and will satisfy values. So.
Okay, sorry for delay of 2 to 3 minutes. We will make up in the coming session. Okay, or we will carry out this 5 minutes more. Right. So now the tutorial actually, as you have seen the previous tutorials also, this tutorial is also based on some experiments, and rather it should be the both ways we can include our discussions, and also we can see that how the things are happening and what type of enzymes you can target, and also you can see these enzymes and their modeling patterns. So there are few aspects of bioinformatics, computational biology, systems biology, metabolic engineering are covered. So I just open the slides, please. Alright. So, as you know that there are lots of techniques which we have described in the previous lectures, but you need to understand how to do those experiments and how to get the various type of data. This one. Okay, so we will go through these slides and see what type of experiments we can plan out of this tutorial. And also, I request you to keep various type of discussions open during the tutorials. Also, you can ask. We will try to have more interactive. Then Dr. K.K. Sharma will also help us in just showing you the simulation videos, how it has to be done. Okay, now you see the modeling of metabolic networks and enzyme modeling. For both things, you need to have the background of bioinformatics. So the students who have, the participants, the faculty members who have no background of bioinformatics, they have to learn the basic principles of bioinformatics first. And also they should have some ideas that what type of softwares we are using for various metabolic network designs or redesign, and rather they could understand the principles only when they know about the database, because most of things are related to computational biology. So it is not possible for you to learn in 2 hours or 3 hours. You can see these slides, you can see the various type of experiments which are done. Later on you can practice, or also we can help you because our department is involved in all these activities. So we can also give you more input about that. So if you see outline of this tutorial, we have introduction to metabolism and metabolic networks, constraint-based modeling, mathematical formulation methods. You have to have the ideas of linear programming. Then whatever the research we are doing and our group is doing, or our department is doing, or as a whole for this, that is the integrated metabolic and regulatory networks. And this is another aspect of our one. Now you see what is metabolism. If you see, Metabolism is the totality of all chemical reactions that operate in a living organism. If you talk about metabolic engineering, so first you should understand what is metabolism. So as a microbiologist, we always learn about microbial metabolism, microbial physiology. So whatever enzymes are produced, whatever biofuel you are producing, whatever things you are doing in a cellulosic ethanol production, metabolism is quite important. We are talking about increasing the production, enhancing the production. So these things require the inputs in terms of the use of metabolism. So the metabolism is basically the chemical reactions. It is whatever is going in your body is type of chemical reaction. In the same way, whatever is done by the microorganism is also done by the chemical reactions. So you have two things in metabolism. One is catabolism and one is anabolism. So you should have, at least if there is a cell, if you see this picture, you can find that this catabolism and anabolism, these two things are completing the metabolic aspects. But when you go to metabolism, the question is there, we are microbiologists, we are enzyme technologists, we are other field researchers. Why should we study this metabolism? And what we will get into it. If you go through this, there are a few things that I say that it has a lot of importance in designing new medicines or new type of medicines. Like you can also understand metabolic diseases, all diseases which are occurring. If this is a metabolic disease, it is due to the aberration in the metabolism. Maybe some gene network or something, aberrations in the gene, or some enzyme machinery failure or something like that. So many things we don't know. Still, biomarker discovery, if you see, biomarker, enzyme-based biomarker. All these discoveries, all this research is based on this particular aspect, that how to have these enzymes, like metabolic enzymes and their regulators gradually becoming viable drug targets. So I think most of the people these days, they are targeting various type of enzymes and then enzyme-based drug design. That is also very, very common thing. So you cannot have a biomarker discovery, you cannot get a biomarker until unless you understand the metabolic part of it. So if you see this thing, this is very, very important. And then it comes to the bioengineering. So you have to have the metabolism study to understand these things as well as the bioengineering, means efficient production of biological product that we are studying since last 3-4 days this week. So if you want to produce in commercial production of ethanol also, you always say that we want to have enhanced ethanol production. So it is possible. It is not a dream now. It is possible. You can do this type of productions by using this type of machinery and also changing the metabolism of that particular microbe. Now, if you see the best understood cellular network, if you are able to understand a bacteria, if you want to understand, it might take 10 years, 20 years, 30 years. Many people, they have devoted whole life in one organism. They work with one organism and they have understood all the things about that. If you see the researchers which are based on physiology, microbial physiology, that is separately they are understanding the physiological importance and the metabolic networks. So this is very, very important thing to understand the cellular network. So why we are studying, this is the logic that you can understand so many things. So one thing, if you target, like if you are targeting this thing, because in this lecture we are discussing about this bioengineering. So efficient production of biological products, how it can be done. So if you see, this is the way where metabolites and biochemical reactions take place. Now I have made this metabolic pathway. You can see this. There are so many types of pathway, but if you see, biochemical reactions are the process in which two or more molecules interact, usually with the help of enzyme and produce a product. So very simple terminology of a metabolic reactions or biochemical reactions. But this is very simple, that glucose plus ATP gives you glucose 6-phosphate plus ATP. Very simple. But how it is given with the help of glucokinase, how the glucokinase is reacting, what are the changes, what type of glucokinase it is, which type of bacteria it is produced or fungi is produced, and why this is like this, why glucose ATP is generating this. These are the questions. And if this pathway is helpful in any type of principles which we use for any industrial products, then definitely it terms that whether this reaction can be modeled or can be controlled or can be enhanced in some pathways. So this is a very simple example where you can learn that okay, this is the example and this enzyme is taking place the metabolic network pathways. So if you want to model this enzyme, you can understand various type of discoveries. Most of the reactions are catalyzed by enzymes. This is truth. And these enzymes, if they are catalyzed by enzymes, these enzymes has to be properly studied. Not only their structure, their purification. We need to understand each and everything. That comes to the tutorial part. Now, modeling, what are the things we have to consider during this modeling of the network function, kinetic models? We can have metabolic dynamics of metabolic behavior over time. These are the prerequisites. You should have metabolite concentration. Work on metabolite concentration. How much metabolite concentration is sufficient to perform this type of reactions? This, like if you are working on bioethanol, we are targeting bioethanol. So how much metabolite is important, and how much concentration is good for good aid. And also, not only some papers you can refer, but you can also try to improve, like with the substrate engineering or substrate modification. So this is important. The enzyme concentration, enzyme activity rate, solved using a set of different equations, and it is impossible to model large scale network because small scale network you can do that. Like if you talk about the bioremediation network, I was reading a paper. So if you talk about that bioremediation networks, there are so many types of enzymes are involved, and not only bioremediation, other aspects also, which we don't know, even plant microbe interactions. If you talk about PGPR related thing or anything which is related to biological network design. So large scale models and networking is very difficult. It is not an easy task because you are not able to understand many of the networks which are within the cells and within the enzymes. And there are many isoenzymes also. You have so many types of isoenzymes. And until unless you don't purify them, you cannot understand what is the role of those enzymes. So this is a very challenging task, but we can do it with the help of these tools and software. So this is impossible, but is possible. But we need to understand these concepts. So because this is a tutorial, so we have to learn that what is need for doing all that. So impossible can be possible, which if you have a specific enzyme rates data, and then it is complicated, so you have to make it simplified. Now, if you see this is a very good slide. I really like this because it explains that you need not to worry about learning dynamic systems, because dynamic system understanding will give you most of the data. But most of the enzyme technology students or students of microbiology, we used to do kinetics of enzyme. Sometime our supervisor is also telling, sometime we have to do the kinetics. Without kinetics, I think none of the enzyme technology will work. So if you see the dynamical system, you understand this kinetic constant purpose. So most of the enzymes where the kinetic parameters are not properly studied or unknown, you have to find it out. You have to design the kinetic models. So if you are able to design the kinetic models, you can have various type of other ideas. So if you see accuracy and scale, if you see one is, if you have these things, you are accurate that this kinetic model is for this enzyme, and this enzyme is first time kinetic model is generated. So it is possible to have accuracy with the help of this, but if you have the graph theory like you do normally, you have in your enzyme studies, kinetic studies, Michaelis-Menten studies, and other studies which are very common. you can have the topological study. So there is a graph theory which gives you structural network properties, degree of distribution, centrality, and clusters. So this is also very interesting. You know, once you start with it, you will learn all that. So I have another tutorial that I will give you tomorrow that gives you real identification of those type of graph theory, specific some enzymes. So this is very good slide which gives you the ideas about accuracy versus and scale. So what is scale you can understand and what accuracy of the models it can be there. Now it is coming conventional functional now. This is most of the researchers they do this because it doesn't require much data. You should have some probabilistic models and some discrete models. So you can get this from here. If you start, you are halfway, just below the halfway of accuracy. But at least the scaling and the scaling of the network and the network function you can understand. But if you generate all these things, topological analysis complete, conventional complete, kinetic studies, dynamical system, everything. So it is complete modeling of the network system. So if you are able to do all these things together, I think it itself is a big project. So if you are working on one enzyme, you can concentrate on that with the help of various type of. Now you do this. These people they have just started working. That is the constraint-based models. You can find a lot of papers in Google and Science Direct about constraint-based modeling, and this is very good modeling. And I have seen few of the models and few of the stories which they have published in the form of some research reviews, and also they have published in some research papers. So this is constraint-based modeling, which is based on optimization theory. So constraint space of possible steady-state network behavior. So basically, they are taking steady-state network behaviors, which are known. There is no other behavior. This is our known behavior. So get the metabolic models from the known ideas, known networks, with the help of software. That is the FBA. You have known about that FBA-based model. So flux balance analysis. Later on, you do that. So I will show you that also. So this is the complete set. And if you see, you can have constraint-based modeling theory, because very important thing it is. So how it has to be done? It is what it provides. It provides a steady-state description of metabolic behavior, a single constant flux rate for each reaction. It also denotes the metabolite concentration. It is independent of enzyme activity rates. It assumes a set of constraints on a reaction flux, and it also has components of genome scale model. It can also generate. So flux rate can be calculated in this unit, and you can find. See, this is basically, if you see, this is the reaction, and the flux. Those who are working on flux balance analysis, it is the complete combination of what type of flux it is. Like V1 is there, V1 is there, V2 is there. So this is a very simple thing. I will show you some of the networks which are very complicated. So those type of network, the software is able to define. We did it with some students earlier, and they also give you the genome scale models. That if you are able to design something with the help of you have the genome map, then you try to see the gene over there and what gene function is controlling that type of flux. So which flux is controlled by which gene. So this also you can do. And the best thing is that constraint-based modeling has it is independent of enzyme activity rates. So if your enzyme activity rate are not properly calculated or not properly, like there are different type of enzymes, especially in bioethanol. If you see, there different type of conditions are coming. So sometimes the temperature is 30 degrees, sometimes 40 degrees. It is thermophilic 50 degree plus. So you have these aspects which you can do. Now, in constraint-based modeling, you have to find a steady-state flux distribution through all biochemical reactions. Under the constraint, you have to do the mass balance. See, we cannot do mass balance ourselves. This is a part of chemical engineering. How? Mass balance analysis. Yes. So it's a part of chemical engineering. So you have to learn, and you can learn from engineering students. They can do it very well. Then mass balance and metabolic mass balance. Under mass balance, you have to do the metabolic production, metabolite production, and consumption rates. Then thermodynamic reactions, that is a part of biochemistry. Then enzymatic capability, then availability of nutrients. These are the constraints. When you generate a model, make a model, these constraints are always there. But this generates a very good model for your study. If you have the data about this, and these data are fixed in softwares, and you can definitely understand this particular part as a whole. Now we go to the next slide. If you see, I will just show you one very good. Sorry, this is later. Additional constraint. Yes, these are the, in the previous slide you have seen. These are the constraints, known constraints. We know that okay, these will be there in any type of metabolic reactions. But if you proceed further, you can find additional constraint. Okay, here the constraint, the meaning of the constraint basically is that you cannot do certain things. There is a constraint. But here constraint is very good. You should have a lot of things which you can consider. Especially in bioethanol production, I think nobody has tried this constraint-based modeling. Very less paper you have. So if you see any of that research which you are doing, this becomes very important. So this is a transcription regulatory constraint. If you see this paper, Untitled 2000, have described this very well. It is not easily understood without the training of your basic training of Boolean networks or Boolean design networks, and also understanding the concepts of this regulatory constraint-based model. So if you see, this describes the Boolean representation of network. Then energy balance analysis, it is described by Beard et al. 2002. It's a very good paper again. Loops are not feasible according to the thermodynamic principles. Then reaction direction, it depends on the metabolic construction. So now you move to this and you see this one. Now you have this FBA solution space. These are the meaningful solutions. Means overall, if you see, this is the whole what we have discussed. Now you have some solutions here as a whole. So maybe these solutions are good for your research, might be they not helpful for you. So it depends on your research, your data, your experimentations, your repetition of the experiments, because all these data, kinetic parameters specifically, you have to run a lot of wet lab experiments, then you get the kinetic data of that particular design. And if you are taking data from some databases, that is also possible for your test. You can do that. So this is the meaningful solution which will give you certain type of. Now you see this very good information here, and this is based on the various type of wet lab experiments. And
sir, how will we confirm that the solution which we have got is useful for us? How will we confirm that?
Actually, in metabolic network design, the metabolic network is constructed, and there is a signal coming. What we have done through the Cobra tools. Okay, so sometimes the tool is giving you the signal. Okay, and some green dots are coming. Then you combine those dots and try to find out if some meaningful solution is coming. So they tell you the probability. Okay, so we can presume it. Okay, and normally when you start working on a research problem, like I myself is working on this BA. Okay, so in my studies, my own project. Okay, so I have started learning this. So we cannot say that okay, this is meaningful or not, but certain type of signals are coming. Then you compare it and try to get some resolution out of it. Then we can see them. So it requires some practical training to understand that. Now this metabolic network, if you see, you can do everything provided you have the knowledge. So like we are biochemistry people, we learn biochemistry. We learn genome annotation also. We also learn the physiology of microbial physiology. Then also we have some reaction engineering or we know reaction. So if we have the background of this, or we have some people who can teach us all these aspects, then definitely you can understand this metabolic network design. And if you see this particular chart, this, and along with this. So all these things coming together will give you the solution for systems biology and metabolic engineering aspects. So it is not possible, and analytical methods, like you should have all the data, related analytical data of that particular enzyme, which is crucial for making these type of models. This is a genome scale models, genome scale networks of reconstruction today. So if you see, these are the references. I will give you these papers. These are few organisms where you have the genome scale networks reconstruction. Reconstruction doesn't mean you are making something very new. So it is just reconstruction is the meaning is that you are trying to build based on your need and your requirement. So your organism or your lab organism or your product which is to be then. So that is the construction reconstruction. That term is used. So earlier I thought that reconstruction means something very novel coming up. It is not very novel. If you read a high impact journal that is metabolic engineering, so it's a very good journal, metabolic engineering, and they have another journal that is metabolic engineering communications. These two journals, at least you try to see one paper each day. Small paper they are publishing, but very good paper. But all papers, they are having the ideas which are generated, data is available, and you can choose data with your available data and try to have a complete system. So this is a very good. Now you see number of genes, number of metabolites, number of reactions, and these are the years if you see. I have not written anything after you see 2004, right? So you don't have any information basically which is coming out of this. Like if this model, E. coli, a lot of people they are working. E. coli, I earlier heard about E. coli, we heard that it is a pathogen. But I read a lot of stories of fruitful products and very important products, and also it is used as a very good host. So many things come. So E. coli, and it's a good model for small biology. So you have a lot of this. Then Helicobacter pylori is a very important microbe. I think you must be knowing about that. This H. pylori, what it causes? Which disease is called? Huh? Peptic ulcer. Okay, so this is also very important. And other, I think there are some type of cancer also it is, right? From Yes. So then Saccharomyces cerevisiae, he talked so much about that. So this is also very good. Then geobacter, then mitochondria. If you go further, constraint-based modeling applications. What applications you can do in your research? You can do the growth rates across media, knockout lethality, nutrient uptake, secretion rate, intracellular flux. I think you just switch it off for some. Intracellular flux, growth rate following adaptive evolution. So, if you talk about a student, like I am a student, I would like to have this knockout lethality as a one experiment for my project. Or maybe I like to have something with measures the intracellular fluxes. So, any microorganism where you are working, then bioengineering, very good term. Bioengineering, I'm in bioengineering part. Bioengineering, there are so many aspects. This is one which I am talking about, is the strain design. Overproduce desired compound. So strain design through bioengineering. So if you are able to control or model the metabolic network and design a strain, like you have designed Bacillus ceteris, okay, MDU 1 or maybe any type of a strain which you have designed through bioengineering, it gives a very good credit, and people will rely to have those strains for their bioethanol production or whatever is there. Then biomedical, it predicts drug targets for metabolic diseases. I already explained you. Studying an array of questions regarding these things. So many things we don't know whether metabolic network can give you answer to all the questions. No, it is not like that. Many people will say that whether it has been done in vitro or it has been done in the cell culture. So you don't have the answer. Even if you ask me whether it is feasible to do in the lab, and it is not possible, and it is not possible without doing. So you try, you see the experiment, you try to get some results, then you compare with the published study. Then if it is a new, it will be definitely a novel thing. So this is one very good data. Phenotypic prediction, knockout lethality in E. coli. Now you see this is the one experiment. This type of data is coming, and these softwares, they are very giving very good data, images, diagrams, which are really very interesting to see at least. Like if you get a metabolic network of any type of constituent or chromatic bacteria or fungi, and like for biofuel production. So I can show you that data, but not today, maybe tomorrow. I will show you that data, but because it is under publication, so I am not showing all that thing here. But it is really very interesting. So we did a model from Aspergillus niger for FBA and all that we have done. So that is very interesting thing. Then phenotypic predictions, flux predictions. So this, if you see this flux prediction, this is also done by the software. So you can predict the metabolic fluxes following the gene knockouts, search for the short alternative pathway to adopt for gene knockouts. So if you are taking a small, the bigger pathway, the flux cannot be calculated as a whole. So you have to see the regulatory on-off mechanism. It is called minimization. So you are taking short alternate pathway here, like see, if you have seen some alternate pathway, you try to focus on that so that you can predict something. You cannot predict everything, but if you are able to predict that, okay, this E. coli is producing this metabolite, and this is the regulatory on-off minimization. This is phenotypic prediction. How the growth rate is evolving. You see this. This is stable. This is the evolving, which is a 700, and you see day 0. You can day 60. So it needs a lot of patience also, that you need to carry out your experiment for 60 days by not taking any leave or not taking off for this. So if you are able to understand, you have to compare it from day 0, day 40, and these are all data which will give you the idea about the evolving growth. This growth rate modification, like you have knocked out certain gene for some network, and you have told everybody that I am going to get higher production by this metabolite or by controlling this flux. So you have to have the data about it, that whether the growth rate is increasing or decreasing or stable or whatever is there according to your need. Now, the strain design, maximizing the metabolite production rate. I think I will see here that if you identify a set of genes whose knockout increases the production rate of some metabolite, the knockout of reaction V3 increases the production rate of metabolite. Here, this, if you see F and V3. So this is one example, just one example. There are more than 100 or 200 type of networks like that. So when you make a network map or metabolic network engineering, you learn that, okay, this is, you see, suboptimal mutant flux distribution. This is optimal mutant flux. Means this is through that machinery that you have find out certain type of pathways, alternate pathways, and you have designed it. If you go further, the mathematical representation I can show, but I cannot explain you much because I have learned from somebody. So this is some mathematical representation. You can see I have taken the same example here, but it can go. So if you see, I told you you need to understand the mass balance, thermodynamic properties, and capacity. So if you are able to see this, means you can understand all the aspects of any enzyme. I am talking about enzyme glucokinase or this pathway. So if you are able to understand or able to design a proper network topology with all the data which you have for biochemical reactions, then something can come out about your particular product. Now this is another thing which is very good for your practical purpose, that is how to identify the plausible physiological state. So optimization methods are maximal biomass production rate, minimal ATP, minimal nutrient uptake, And normally the enzyme system or whatever, you have nutrient uptake and then how much biomass production is there. Because especially in bioethanol, biomass is very important. How the biomass transformation is there, how much recovery is there. So this plays a very important role. Then exploring the solution space, extreme pathway and elementary models. So I'm not going on detail that. This is one example where you can see the metabolites and you can have this demand. So this is also published. This was published, and you can see this paper. You can find a metabolite and how much demand. So if you see various macromolecules, you can work amino acids, carbohydrates, ribonucleotides, deoxyribonucleotides, sterols, fatty acids, lipids. Okay, so you can work on these. These precursors are removed from the metabolic network in the corresponding residues. So the growth is defined as like this. So if any precursor is there, means you have to think about that if any metabolic network study has to be done, the enzymatic behavior, enzymatic recovery, then precursors, then coenzyme, isoenzymes, so many things are there. So then FBA. So FBA, I will just explain you a few things. First, if you see this paper, Peloton, it's the first paper related to this particular thing, and Verma and Carlson. This Verma is of Indian origin, but not in India working. But they have done very good study, published in. It's a very small paper, which gives you idea. So if you see finds flux distribution with maximal growth rate. So biomass production rate represents the growth rate, and it is solved during the linear programming. So if you go further, this is one example, okay, where you can see. So if you see, this is the H externals and this is the cell membrane. Now if you find this R1, R2 and the gene, okay, each gene is responsible for different type of enzyme and same type of flux. So if you see this, it's a very simple thing to explain you. And most of the people, they believe on one gene, one enzyme theory. It is same like that. But there is another thing, one enzyme, one gene, one enzyme, one flux. So one flux and one metabolite and one, so many things will come in. But even if you are not able to understand one gene or two gene or three, here the benefit is that you can understand all these genes in detail. But you have to do experimentations. And this is the FP example too, where you are having all these vectors made, okay, and also get idea about various type of genes which are responsible for the various type of flux which are generated. A very interesting thing. And there is a pattern. If you see, we are not the mathematicians. There is a pattern here. Okay, so that pattern also has to be followed by that particular cell. Now this is the mass balance and this is the flux constraint. So you people will ask you to generate the mass balance and flux constraint. What is the objective function, and then z is equal to b growth. Hence you can have this calculated for your metabolite. This I'm not going in detail because this might take more complications. So I'm just excluding this. I will give you the writer because you have to do the linear program, which is not possible for a microhealth to do that, but you have to take help from a chemical engineer and they can help you in this. And just go further. Okay, this is topological methods. It is not biased by the statement of an objective. So network-based pathways, extreme pathways. If you see this paper, elementary flux model designed by this Suster 99. And if you see, you have to have the random sampling data for all the microbes which you are working. But in case of metabolite like bioethanol, you can either go for the microbial means fungal system or bacterial system. So whatever system you are working, fungal or bacterial, you have to take the network and the biochemical pathways which are assigned for the production of bioethanol. And these are few elementary and reaction networks which are giving you the extreme pathway. So we are focusing on integrating the metabolic and regulatory networks. And these are the constraints if you see. So FBA, now coming to FBA, it predicts that both galactose and glucose are simultaneously consumed when present in the media. So when glucose is present, the concentration of active CRP decreases and represents the expression of gas system. In the same way, Boolean logic formulation will be like this, and then you can have the regulatory state and metabolic state. And this is the metabolic regulatory network, and this is the metabolic network. So if you see, you can have enzyme 1, enzyme 2, this is a complex, and they are controlled by various type of genes. And if you see genome scale integrated model of E. coli, which was proposed at 1010 genes and 817 proteins and 1083 reactions. Might be more reactions are available right now, but we cannot say that is the limit. So if you are able to generate a genome scale model for any microorganism, especially for phenol production, you can definitely do that. But you have to learn the things which I have presented earlier, and also learn this regulatory state understanding, so that you have all the ideas about this. So research objectives you can frame is that you can generate a model, or also you can have the study the expression and metabolic activity profile of genes in E. coli. One example you can take. So I'm not going in detail about that. It is something. Flux prediction. Okay. This is also very important. The prediction of flux, metabolic flux. You can predict it, and this is all done by the software. Okay. So you can have these through the data via NMR spectroscopy, where you get the data from there and then you update it, anaerobic and aerobic. This is all about the flux balance, flux predictions. And then in detail, because this is something, okay. Okay, these are few genes where you can find this validation of expressed genes. You can find, if you see the summary, you see we can develop the method. We are still working on that. Also, we can identify 36. So, if these genes, these are already identified. Some of genes controlling the biofuel we can do. And one thing which is very important is that 12 genes, this is identified by some other groups, they are able to predict it to be non-expressed. So if this particular aspect, any media, anything, I have not seen any good paper on that it is controlling all those aspects of metabolic flux or something in biogenome. But I think we can see online, and we can search some new paper which we can discuss in the discussion session. So we'll go through it and learn more of that. So I think this is all which can comment. Now I have not explained much about the modeling which I showed you in the beginning of the class. So some of the modeling and tutorials about the simulations will be explained by Dr. K. Sharma. So I invite Dr. Sharma to take over the session and explain the basic thing. And I will I invite Dr. Sharma for the further. How much time we have left? 30 minutes? 40 minutes? Okay, 1 hour we. So because we have to discuss also for another 30-40 minutes. So I think Dr. K. Sharma will take some time, and he has very good simulation results from his lab, which he will present and explain us also. Yes, stop, stop. Good afternoon, everybody. It's not a lecture, but experience which I had gained. I'm not a system biologist or plant phonetician, but I learned these things after my PhD, and I was having some interest. So while doing PhD or doing masters, but those days we were not having such tools and softwares. But and some of the data which I will show you, it's a story. It's not nothing like a lecture because it's a tutorial. You know, it's a tutorial. So it's a story. I'll start with one story. And I discussed the story with my master student also during life history classes. It's a simple story, and Dr. Shukla wants to show the video. So the panel is open for discussion anytime you want. You want to discuss, you discuss with me. I don't talk anything very scientific, very technical, but those are concepts you want, you can come to my lab and you can handle the software, work with the software regarding what you want to make a model, you want to copy, you want to simulate, you can do.
all the softwares are lying there, and you are free to do that. Story is very 2-3 stories I will share with you. We earlier we started making models of Yersinia laches. It's a Yersinia laches. Why we made model? Because there, if you take any system which is not well established, a wet lab system, it's not established in your lab, then obviously you will go for something which can be easily done on computers, like in silico. We cloned, so for that what we did, we cloned laches from Yersinia interproticus, different strains of Yersinia interproticus. We cloned laches and we made a model to find out what laches does. Because there were no report of laches, no report of laches from a pathogen. In year 2003-4, there were no report. So we cloned, and after clone, we instead of going for expression studies, because this expression studies is very challenging, and we need and what to do after expression, because after expression you have to do a lot of wet labs, and you have to do a lot of interactions with like metabolic studies that I was talking about. So we don't know what lattice does in Yersinia. So you have less very short time, and you have to screen so many metabolites with this lattice new protein, which you had reported. First time we reported this lattice and published in PBRC. And second work was also published in PBRC because it was a novel. It was the first report of Yersinia. So we made a model and we choose different ligand based on the idea, because first you have to make a hypothesis what this lattice might be doing in Yersinia or in any pathogen. Not Yersinia, I consider Yersinia as a pathogen. So any pathogen. So when a patient suffers from diarrhea, recently Yersinia is also reported for IBD and Crohn's. So when a patient suffers from diarrhea, then in that case he goes to a doctor and he takes some painkiller first. Some painkiller, and he needs something to stop diarrhea. And when he takes painkiller like diclofenac or aspirin, anything like diclofenac, I started with the diclofenac. So how does that with a dysbiosis, having a lot of microflora which is different from normal, how will it behave? So what we presume that lactase, we know it is a multi-purpose polyphenol oxidase, and it has the property to degrade any xenobiotic compound. So considering a xenobiotic compound, we took first consider a lot of xenobiotic. Even your dyes are also there. I will show you some data with dyes, industrial dyes. Dyes are also xenobiotic compound. Pesticides is also xenobiotic compound, and which is not yet human. So your diachronic is also different. So we made a model and we choose a lot of painkillers, lot of other drugs, and we try to block it. Modeling, we made model with the help of modeler and docked with the help of DC Gold. You can also dock with the help of other like Autodock. You can use Autodock. People are using Autodock very commonly. But we have DC Gold. You can use any software for modeling or for information. You can use any software. Only thing is you need to validate your model and then validate your docking experiment. And after validation, if you could simulate it, if you can mimic the experiment experimentally how it might be happening, that will further validate your data, further improve your confidence on the data. But when we made a model, docked and simulated, docking we got result, but when we simulated, we didn't got the result, proper result. What happened during simulation? The ligand flew out of the active site. This was devastating data. Means modeling, we could have published modeling and docking. We tried to do simulation, but to further validate the data so that we could get some authentic, we could further confirm our result. But result was devastating. We had collaborated with Professor Andrew Lane in JNU, and the student told sir, your diclofenac, aspirin, they are not binding to the active site. They are coming out of the active site after a few nanoseconds. So what to do? because docking experiment showing good result. Because when you do, there is a constant of in silico experiment that it has to be done in a single parameter. You take pH 7 as a standard parameter by docking. See, pH has a very great role to play for binding any layer with the enzyme or with the protein. If you change the pH, the binding efficiency can go up or down. So it was going out of that. Then I told, can you do something in silico? Can you change the pH hydrogen ion concentration in silico? And he told me that somebody in our lab, she knows how to do this, and she made a small script, and we could change the hydrogen ion concentration around the active site, so that because it's hydrogen ion concentration proton, it's a proton which more the proton is, lesser with the pH, higher, lower the proton is, higher with the pH, it will be alkaline. So with the changing in proton concentration and then doing sublimation, we got very nice result. He called me and told, sir, at acidic pH 5 and 7 neutral pH, it was not binding. But when we did the 9 pH, the binding was very strong. So this is the beauty, this is the power of doing in silico work. That without doing any experiment, we came to the conclusion that aspirin or diclofenac binds with the protein at alkaline pH. So when the pH changes, so it means that it is metabolized in the intestine rather than stomach. It will not be metabolized in stomach because lactase and yersinia, because when you have dysbiosis, you have abundance of dysbiosis, you have abundance of this bacteria from Enterobacteriaceae family, and most of the Enterobacteriaceae family now even we have reported has lactase or multi-complexes. Most of the Enterobacteriaceae. So what does this lactase do? क्या करता है? It might be having some role to play. What we have found, it actually is a redox enzyme, and it helps in redox signaling. It does redox signaling, and it communicates with different microflora and with the intestinal redox enzyme, and it changes the whole redox environment. It is actually a way of communicating among different microflora during dysbiosis. With this background, I will show you some videos of simulation, and because if I go on talking about the experiments, and because it's a tutorial, but I will show something practically how we did and what is the strength of this molecular model, required homology model, because it's based on homology modeling and docking. So this is, I will show you some videos, and I won't tell you about the pH on which pH it was performed, but you can guess yourself. See, this is a simulation work. This one is here. So this one, you see, this is a copper, central copper metal ion, and you have a ligand here. See, it's not going out. How does it make? It is made with the help of movie maker. What simulation does? It is for 25 nanoseconds. How we make this? We check all the combinations a ligand can make, all the probable bonding a ligand can make with the enzyme, all the probable bonding, and a snapshot is taken at each picosecond. This is a 25 nanosecond. Each picosecond we take a snapshot with the help of we had used Promax software, online software, and you get more than 1,000 of snapshot, and then we compress all those with the help of movie maker. What you do, we do a cascading and all the still photographs are transformed in the form of videos. So and modeling with metal protein is not so easy also, because you have to incorporate metal add after you make a model. That is very complex. Doing a modeling experiment with non-metal protein, it doesn't have any ligand, any central metal atom, any cofactor is easy. But putting any, if you want to put any metal atom, it is very tricky. It took more than 8 to 9 months. My new student, she tried for 8 to 9 months to put, but we didn't got the success. And one very strange part with this, which I have faced with this field is modeling blocking people. They will never reveal how they do. What I have read, they will never tell you how they inserted the copper. They will never tell how they have changed the pH, what steps they had written. They never share these things, because they think that if somebody knows these things, because it's part of tutorial, that's why I'm saying, they may do it, because this is a very fast science. You can sit on the computer and do it yourself. If you know the trick of the trade, you can do it. You can publish a lot of papers in very short duration of time, because genes are there, PDB database is there. You need for homology modeling, you need just a protein with good resolution. Somebody might have done it, this crystal, they have made a crystal and they had done diffraction, pure diffraction data. You just need a protein, you just need a gene, not even protein. You just translate it in silico and you can develop a model. That is the beauty of this modeling experiment. I will show you another one. I'll show you all the video. Yes, it's. See, first one was at pH 5. Second one, this is at pH 7. That was pH 5. This is at 7 pH. Next one. This one? Yes. This is at pH 9.
And pH
9. 9. See the difference where it is binding.
you change the pH.
Yeah, you change the pH.
Yes.
It is very far away from the metal, copper metal.
Can you change the temperature?
You can do that, yes.
And solvent?
Solid also, everything. Yeah, this will get strong. Now, I'll show another one. See, I think this is another one. See this one. This will be very interesting. So,
What does it mean?
it means that at pH 5, this is another one that was a set of 3. pH 5, pH 7, and pH 9. We saw all the 3 different pH, and the ligand was ABTS. I didn't reveal the ligand first. I didn't reveal the ligand. What was the ligand? That was ABTS. And that is a confirmed substrate for lactase. Lactase, fungal lactase. And it was binding with the bacterial lactase. And this was the first report. We even checked before doing the experiment with wet lab experiment with bacterial lactase, because we thought that at genetic level we are getting lactase. Maybe those lactase might not be functioning in seed. Maybe. Because at gene level, through gene scan, we got lactase first and we got lactase through gene scan. We scanned gene from different pathogens, even from mouse, human, and you're surprised we got lactase in human, mouse, and your archaea. First time we put archaea lactase also. Archaea, and then Yersinia pestis, Yersinia epilotica. Everywhere we were getting lactase. It is universally present now. What I feel. It has some role to play in redox, balancing redox, disbalancing, rather than we can say disbalancing, because if there is abundant of this Yersinia epilotica or lactate, so more is the micro, more is the protein, because when it lies, it will. And these lactates we had done biochemical characterization of their stable at 9 pH, 8 to 9 pH. They are optimal at 8 to 5 pH, 8.5 pH, and at high temperature also they are very stable.
So this is the salt. For example, sodium chloride.
Yeah.
In lab.
Yeah. Yeah, it can be done. So the environment has to be changed. Here environment we are keeping water. Here we are keeping water. And sodium chloride, during sublimation we are keeping ions. We are keeping ions. We have to maintain the ionic environment artificially in silico. We have to keep the ionic environment. So we are keeping.
What is the user to do it? You use Bromax.
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 MDBPSA. It's a MDBPSA. MDBPSA.
PSA.
It is a software which is in Bromax. It's done by Bromax.
Bromax.
Yeah. Bromax.
So it is software.
Software.
Based on Bromax.
Bromax is based on Bromax. So this was at pH 5, and the structure was aspirin. This time the structure was aspirin. So you will see that in the case of ABTS, which is a true substrate for lactase, it is stable at 5 pH and 7 pH. 9 pH it was a bit far away from the active site, but it was not out of the protein. It was at least not, but aspirin, which is a painkiller, it blew out of the active site. It blew out of the active site. That means it was not at the active site. It means that at 5 pH it can't transform the lactase, because for any transformation or any reaction, any metabolic reaction, prerequisite is it should interact with the active site, around the active site. It has to make some hydrogen bond. So you see, it means that at 5 pH it won't transform. So whenever you're going for a rat model study, this is not actually. I'm not supposed to talk about the experiment here, because it's students. Otherwise, I won't talk to you regarding. We have confirmed these experiments at rat model also, and we have seen that at different pH conditions they have different effect. So because we have to, for the after summation also have to go to some animal model to check. So very easy, very convenient one is that model. So now we'll see at pH 4, pH 5 we have seen for aspirin. Now this is at pH 7, aspirin at pH 7. No, it's not binding at pH 7 also. It is out. So it should not bind at pH 7, no reaction. Let us see at alkaline pH. Again, aspirin at alkaline pH. It is there. It is near the active site. It's moving around the active site. This is at alkaline pH. It means that, and these are validated in wet lab. This means that aspirin can be transformed at alkaline pH in this small intestine or large intestine, not in stomach. It can transform, and when it gets transformed, it has devastating role. Most of all of the aspirin or all of drugs are not metabolized, and when they are not absorbed, and when they are metabolized into some other byproduct, those byproduct has devastation role. Why we took aspirin and diclofenac? Because it is well known nowadays that these painkillers they cause perforation in intestine. So the abuse of this drug, because in rheumatism or other severe disease, chronic disease, we take drugs without consulting a doctor, and we take this drug painkiller in routine to combat the pain in routine. And abuse of the drug will cause perforation in intestine. That's why, and it's not just aspirin. What we have seen that, what we have to say that it's the metabolite, which is actually the byproduct of aspirin or byproduct of painkiller. It's not known what byproduct they are making and how they are making the byproduct. Actually, through transformation only, and they are transformed through some of the processes or some of the, like copper, that is like protein. They might be transformed. So that is the main idea behind why we are working on racina lactase, why we took some of the model drugs, because these are the molecules, these are the ligands which might be interacting in vivo. So we have to put some system to start with. We have also studied with certain prostaglandin, because prostaglandin is basically the mechanism through which painkiller works. It blocks the pathway. So we did all this through by using this strong tool modeling and docking tool only. With this tool, we could further actually screen out the system to continue with, and then we go on the rat model. Because of these tools, we could now get approval for rat model study. Because ethical committee, there are ethical committee, they won't give you just to inject anything in the rat and just sacrifice and do the experiment. The committee says you can't sacrifice rat without having preliminary work. You have to report some work. I showed the BBRC paper to the committee, and the chairman was Professor Shravan Singh from AIMS. Now he's director of Bhopal. He was very strict. All the ethical committee members are very strict. Very strict committee. You won't get a single extra rats for experiment. If you tell 50 rats, they will give you 20. If you ask for 100, you will get 40 rats. It's very strict nowadays. You can't sacrifice, because earlier people were just taking rats, especially pharmacy and other, and just injecting drug, and just after injecting they will see what is happening without having any preliminary experiments. So these in silico works help you to at least save some life. Otherwise, what we would have done, we would have experimented at pH 5, pH 7, and then 9, and 5, all the rats they will sacrifice without knowing why they are sacrificed. And we won't get any result, and we will be confused why they won't get any result. It's aspirin, simple aspirin. It should go and it should do something there. It should bind with the protein and it should be transformed, but it won't be transformed because it's not binding. So this is a question. Now you see the next one. I've shown it at pH 9, it was binding. This was aspirin. Now you see another one, it's a diclofenac, again, very strong painkiller. Is that pH 5? pH 5.
So what is the substrate? What is the ligand?
Ligand is diclofenac. Diclofenac. Second painkiller. So diclofenac at pH 5. Now, let's see. It is behaving differently. At pH 7. It's very far from active site. It can't be transformed. It is in the scaffold. It's not in the active site. See, it is in the scaffold.
Scaffold.
Yeah. No. But the aspirin was totally unutilized at pH. Now at pH 9. See, diclofenac is behaving differently than aspirin. You can see that pH 7 was optimal for diclofenac, not 9, but for aspirin 9 was optimal. For diclofenac 7 was optimal, not 9. 9 you can see it is. See.
I have one question.
Yeah.
It's like this.
See, it is out.
So at acidic pH, for example, pH 4, pH 5, okay, you use a ligand and an enzyme to the simulation. Okay. For example, the ligand is phenol, a glycol. Okay.
Yes.
Glycol is very
Yeah.
okay. And at alkaline pH, for example, a pH 10, pH 9, okay. And at alkaline pH, the glycol is a phenolic group, okay, the proton move the proton loss. So can it be simulated in this process?
No, it has limits. You can't you can't tabulate something which is continuous. You have to keep a fixed environment. Yeah. You can't make it continuous. Like if there's some change in pH and
It cannot be changed.
no.
But
But see, because in vivo, the charge, the proton, the charge is not constant. It's keep on changing depending on the reaction. So the proton is increasing or decreasing. So here you have to fix the proton either for to maintain the acidic or alkaline or your neutral conditions. You can't change it. You can't make it continuous process. Yeah. If you have any query,
very good.
you can ask. If you have any query, because I can show you there are a lot of things, but it's not a lecture. It's just my pen drive. Whatever I could get from the pen drive, I can show you. Because Ursula just told me to show some videos.
So
Not lecture.
if you do this in silico simulation, okay, for example, you change the 5 to 10 amino acids,
Yeah.
and you can use this one.
Yes.
This method to check.
Yes.
If it works with the binding at a higher pH. Am I right?
This now it can be done. So one very interesting work we did with Vibrio cholera. Vibrio cholera has evolved. Now they are resistant to ciproflox. Ciproflox, it has resistant to ciproflox, and there was a outbreak of Vibrio cholera in Maharashtra, and some of my collaborators, he was telling he wants to confirm the mechanism. How Vibrio cholera is getting resistant to ciproflox. My collaborator at GIAN, and now he's presently in AIMS. And they were doing better experiments, but there were certain things they want to generate a model, and they want to do some interaction, DNA protein interaction first time. So these are all negative protein interaction. That was first time they asked us to do some DNA protein interaction. So we did DNA protein interaction for 50 nanoseconds. We didn't perform for 25 nanoseconds because we were doing some experiment which is of that great impact that nobody will rely that you, because earlier the people used to accept the paper at 10 nanoseconds also, but their standards are increasing. Now they are asking 50 nanoseconds. We published with 25 nanoseconds also. We performed that experiment for 25 nanoseconds, and we had proved that Vibrio cholera has evolved a molecule called QNRBC. QNRBC. QNRBC is a molecule to which ciproflox goes and bind and gyrase. So it goes and bind to QNRBC. Because of the binding with the QNRBC, that site is untouched. The antibiotics is not binding to the gyrase because QNRBC is engaging the drug. And we have proved this one, how it is binding to QNRBC, how QNRBC has evolved. They evolved a molecule called QNRBC. It mimics the active with the gyrase. It mimics the gyrase. So this is how they are evolved, and it was proved through this docking and sublimation. I was surprised. It's a clinical biology infection, very good journal, fact 7, clinical biology infection, and they couldn't reject this paper, and they told that we want to publish, but make it very short. They want to publish, but they don't want to publish everything because they don't give 7 page or 8 page. But they ask for to publish it as a data to editor or something like that, very small, 3 page, 2.5 page. And the data which they accepted for publication, they didn't do any wet lab data for publication. I have a paper in my lab. You'll see. I can't find it. It might be there in the pen drive box, but it's just random data. I don't know where what is lying. So I will show you the data, 1 page data, and rest 1 page is text. And data is of simulation only. And what we did, if you want to publish simulation data, it's an online video also. Otherwise, there is a snapshot of different stages of simulation. There can be snapshot of different stages of simulation, which can be published. So it will show that at certain nanosecond it is binding here, at certain nanosecond it is binding somewhere at near the active site, far away from the active site. So that is the only data they told that you can publish 1 data and rest text. And all the 10 to 15 pictures they took. All the pictures are in supplementary, but only this one because it was so attractive. So this modeling docking experiments and the data are very interesting. You can answer so many questions in a very interesting way. It's very catchy data, very informative also, very informative. I will show you one more. Almost time. There are something. Recently, we published again another work in computational biology and chemistry, where I will show you. So this is the picture which can be published instead of putting a video. Video is supplementary also. So these are snapshots which shows the binding of a ligand with the active site at different stage. And so there are thousands of such snapshots which are compressed into movies. Actually, whatever movie you have seen, it is actually compressed data. It is actually created with the help of thousands of such pictures which are taken, and for your information, submission takes like that 50 nanosecond submission took 1 month, 40 days. 40 days your system has to be switched on. If there is a power cut, again you have to start. So 50 days for 50 nanoseconds. It has to be. It takes a lot of time. So for one submission, if you want to, like we have done how many? 3, 6, 9 submission. You can imagine how much time it takes in submission. So if it's take, that was for 25 nanoseconds. That one, which I have shown you. It takes around 10 days to 15 days in one submission. So see the time, how much time it will take in simulation. So it calculates all, it's actually based on the thermodynamic principle. It calculates all the thermodynamic, it generates all the thermodynamic information like free energy and entropy, enthalpy. So on the basis of free energy, especially free energy, it actually looks for the minimum free energy. So if you look for minimum free energy to interacting to each and every atom. So for any optimum binding to be reported or to be confirmed, the ligand has to, each and every atom of ligand has to interact with each and every atom of the protein. Each and every atom of the ligand has to interact. If it is done manually, according to Levinson paradox, it can take billion years. If you take 100 amino acid protein, it will take billion years for it if you want to interact each and every atom with each and every atom of a ligand with each and every atom of a protein, because it's very tough. You might have studied in protein folding, how long it will take if you do manually. These things are done through strong software, very high power software. Moments is not very, it's very strong software, and it is not user friendly also. You have to learn Ubuntu, you have to learn how to work on Ubuntu system. It's not Windows system or DOS system. So you see, these lactase from Topological new problems. It is not that we recently we have published this one in Functional Biology University, where they had reported the interaction of lactase from Topological new after cloning and after expression and other validation was also done, but binding of prostaglandin and serotonin with Topological how it binds with lactase. And so-called neoproteins is a vascular message east, which where Peter Williamson group has reported the first report of pathogenicity of lactase in yeast. This first report, from where I got idea that bacterial lactase might also be having some similar properties. And so this we got a culture from him. They work on you can't culture this because it's daily pathogenic. It is normally isolated from pigeon excreta. Pigeon excreta has a lot of protocols problems isolated from there. But it's pathogenic. It is very deadly for immunocompromised patient. For immunocompromised patient, it is very deadly. It will not work if we are immunocompromised. This is one. See, these are another snapshot of this is of. This is of prostaglandin. This is of serotonin, and this is of phosphatidylcholine. So these are all three different ligands, and can see the. And how do we come to the conclusion that which binding is perfect? That is based on the binding scores. Binding scores. We have to go for binding scores. Higher the binding score, better is your interaction.
For example, in the first picture, the ligand finds there is a site, active site, or close to the, for example, G1 comma. Am I right?
See,
but sometimes it has some other binding,
yes,
and maybe the edit is
different.
Yes.
Yeah,
Let us go.
Actually, actually, it is what has been proposed that enzyme never binds to the active site. Binds close to the active site, not exactly the active site. Yeah, not exactly the active site, but very close to the active site. Close to the active site. You might have seen the link dot of this very clear article. Link dot was clear, the actual bond and everything was clear.
Nearby.
Nearby. But here what is we can see is, and what we could say that it is actually checking each and every interaction. It is not the optimal binding. It is not the optimal binding. There can be thousands of interactions. We can't put thousands of pictures here. You can't put thousands of pictures here. These are just a sample pictures of snapshots. These are just sample pictures. Just we have chosen a few pictures. It's a snapshot. There are thousands of such pictures which are stored. Your system is completely filled after submission. You have to store it. You have to keep it somewhere after one submission. So there are a lot of data which is generated after submission. Lot of data. So these are just few pictures, just representative pictures for one interaction. Yeah, for one interaction. This is just one interaction. You see, if you interact in the movie, you have seen if you interact with each and every region, very fast.
If there is another site, another interaction, you will have another set of pictures.
I have another pictures, but we can't put all the pictures.
But how do you compare that? How do you compare that?
With the help of a movie maker software.
So, movie.
Movie maker software. I will show you one interesting video. I will show you. How movie maker works. See. See the video. Leave.
Yes.
That's another non-scientific output. It's because of talking information and knowledge which I was having. This information was applied in my photograph. Wait a minute, I'll show you this one. See. I can explain docking and submission with the end of this picture. One. This is a flower in my house. Every year I used to, it used to bloom. It is there. And this is what I did. I took around 450 snapshots, 450 photographs using my camera, digital camera with a macro lens, 200, more than 450 photographs, and you'll see a very interesting. In between, you'll see a honeybee coming out of that. That was a bonus which I got because I was not looking for honeybees. It was actually I was just what I just wanted to make a movie. How flower blooms, bloom a flower. You can't take a video. You can't wait for so many hours. It will be very big video. So what I did after every 10 minutes, I took one video. I took one picture. Every 10 minutes, one picture. Every 10 minutes, one picture. It was on tripod. Every 10 minutes, one picture at same location. Luckily for what I saw, when it was about to open, insect came out of the flower, and then I became very energetic. He pushed me there. This is what I never expected. Insect coming from a flower, and then I took another around 150 to 200 pictures again, and then with the help of movie maker, all the pictures were compressed, and a video was generated. This is how talking transformation is done. So you take a number of pictures, you see all the calculations, do all the calculations, and then you compress it and a movie make. This is how a movie was made. Yeah. It's very clear that they are not. This is not a movie. It has been made, and the photograph was taken for 8 hours. I took photograph for 8 hours, and 8 hours have been compressed in just 30 seconds. 8-hour pictures have been compressed in 30 seconds. Likewise, a 40-day simulation is compressed in nanoseconds. 40-day simulation is compressed in nanosecond that we see. This is a video of your simulation. Anybody wants to ask?
Any questions?
This is the data of my each student, and I'll show you something. This is a whole PhD. Whatever I am getting from my center, I am showing you. I don't have anything. I'm not prepared anything, especially for
The DS negative.
This is again snapshot of molecular simulation of passive experiments with Rosebank model. This is another application I did, we did, my student did. This is the application of simulation docking in validation. So we took a lot of dyes, industrial dyes, and it was known already in wet lab that practice has a property of degrading dyes. And we tried to study the efficacy of the enzyme and the pH of the enzyme, everything. pH of the environment in silico without doing any wet lab. This is what we got. See, this is the data which you get after simulation. This is for RMST data, RMST graph. This has to be very much stable. So it is at 25 nanosecond. It is called 25 nanosecond. The stability has reached. It could have been done for 30 or 50 nanosecond to show the stability. We couldn't do it because it takes a lot of time. But you see, initial why we can't stop at 10 nanosecond? Because it is not stable. It takes some time for stability. So see the RMST data, RMST graph, and these are the data which we need to generate to confirm of the simulation. The van der Waals, energy, electrostatic, SASA, reaction simulation, binding energy. These are the data which are further used to validate the efficacy of the unity of the data. The data is work on.
My question is, you can compare the binding scores. Which one is better?
Binding energy.
They are different. Okay.
This is for bacillus femoris. This is for Yersinia.
Yes.
Yeah. So binding energy, more is a negative, it's better.
So what do you can learn from the?
Yes, Yersinia is having better. Yersinia lactase is better than bacillus femoris lactase. Yersinia lactase was better.
Then for this.
For biodegradation. For therapeutic diet.
Okay.
For therapeutic diet. And we have also seen that its pH stability and temperature pH is also stable at very high alkaline pH.
So this
Binding boards
is that generated
from the simulation blocks.
and this first comes into the pictures, right?
It came. This is the output of the Gromax.
Okay. And show me the pictures.
Yeah.
The previous.
Previous.
So the data originates from a sample of the futures languages.
Yeah. These are.
This shows the binding energy at the side.
It shows the binding energy of the optimal binding site.
Optimal.
Yeah.
But,
Then it's not. Here, this one is not optimal. This is the binding energy of a whole Gromax. After whole simulation. Experimental. What's this? Experimental binding energy. Whole. Whether
so you tell us that the ligand has a higher affinity to the right side.
yeah.
Less affinity to the
Left one, bacillus femoris. Actually, bacillus femoris was known for validation. That case is well known. So we took this one as a standard.
So the right side is not maybe not the active site. Yeah, maybe not. Is it? Does it matter?
Yeah, it doesn't bind to active site.
It is a they can bind at another site. Actually, it cannot be.
Yeah, exactly. If it binds to other side, in that case, your RMST data will not be stable. You have to compare this data with this one. You have to also check the
The
serum.
binding energy actually is the everything in molecular dynamics simulation. Yeah, and every this molecular modeler, we say ourselves as molecular modeler, we always try to see that because without that, like they will take this model Y Theoretica, strain 7 minus at least 187. That is always a better model. Okay. So when you talk about the Ramchandran plot also, so during the project and all those studies, they already do all these things. And finally they come out with this. And they simulate only because molecular dynamics simulation is also time taking and
Yes.
rigorous exercise.
Yes.
You know, one simulation is running many times test run.
Yes.
So especially for because I asked him to present his some of the data. Because if we are able to apply this thing in your biorefinery or with the yeast, okay, we have not done anything with yeast or something. So definitely we can have a better picture.
That is the data after validation, after in silico validation. They give the experimental, and these are our experimental results. It shows the efficacy of the lacase from Yersinia. These are recombinant lacase from Yersinia. You can see how it degrades. Yeah. So your rosebank,
What's that?
it's a rosebank one. A is a rosebank one. B is component. C manufactured.
Right.
So your test and transfer. One is confirmed, second is test. So you can see these two are related.
How long it takes to for
It takes maximum 10 seconds. Maximum
several days?
10 minutes. 10 minutes. The grade from 10 minutes. But minutes without minutes. Without minutes. Order it is not. It is published. It is very fast. Yes, it goes very fast.
Anyway, how we have few queries before we finish this session with you. Okay, and the first thing is that like we have completed this co-culturing. Okay, but I think in co-culturing, last time you introduced us about the concepts of using the consortium. So co-culturing, have you tested in your laboratories the
Yes.
co-culturing concept? So how many cultures you have used? Bacteria and yeast combined or yeast different strains
Yeah, different experience.
of yeast?
I often use the Cellulosic and the pig. I have the two strains in my lab.
Okay.
Yes. We do the co-fermentation like this. We use the whole slurry.
And you are doing shake flask or a fermenter?
For example, if I use the corn store, corn
Okay.
store is a mix of corn stalks and corn pods together. And I do the filtration first. It generates spent liquor. In the liquor, there are some simple sugars derived from cellulose. And I do not do any separation at all. Use the mix slurry, but I have to do the then the pH to be 7, around 6.
Actually, co-culturing itself is a big challenge because so many types of culture you are mixing.
We use the commercial enzymes and the two yeasts. One is
Actually,
ferments glucose, another ferments xylose. There's a lot together. But there's inoculate the yeast at different heights, not simultaneously, different heights. 8 hours for hydrolysis, maybe 3 to 4 hours for the Cellulosic, and then I inoculate the
No, what I was saying is that the fermentation temperature.
Fermentation temperature around 28. I always use 28. Because you know in Guangzhou it's hot. And the air conditioning in my lab is around 25. 25. And in the incubator it rotates. And the temperature is slightly higher. Around 28.
Okay.
But during the first stage, I will use elevated temperature around the 40 to 45 degrees. The superficie I produced from Android is okay. You can work at a high temperature. For example, 37 degrees centigrade, it works. But the optimal temperature is still around 30 to 35.
So you make it optimum for the co-chemistry.
If the temperature is high, the glycol, the byproduct, the glycol byproduct is high.
And whatever commercial enzyme you are getting, that is from China, made in China.
Yeah, all of them from China.
All from China.
Only the enzyme. Sometimes I use normal enzyme for the research.
It means the xylanase, how much will be the cost of and xylanase?
Xylanase is very cheap in China.
Very cheap. How much?
How much? We have two types of xylanase. One type is produced by yeast. The price for kilogram, 18 to 25 yuan, Chinese yuan, kilogram. It's very cheap. 18 to 25 Chinese yuan per kilogram.
Per kilogram?
Yes, per kilogram. It's not a big problem, but cellulose has a high.
Xylanase is 18 to 20 yuan.
Xylanase is cheap.
Okay.
18 to 20.
So who are producing? Your company or university or
One of my collaborators.
and you are selling it?
Yeah, sometimes I help them sell the enzymes.
Okay, so government means they have allowed to sell all those enzymes without any restriction to.
Yeah, our collaboration is highly supported.
Okay. So
Always.
that is a good thing. And the students are also working in that. They must be
It
getting.
promotes the GDP.
Oh, GDP. Okay. That is good. And other enzymes, which means your university is also selling all those products.
Yeah, one of the most important enzymes in my lab, I produce the enzymes for a particular purpose to remove the algae.
Okay. Yeah, I saw that.
Contaminants, in Kinetic River. My enzyme works in the sea, in salty water, and in the river water. We tested for 20 times in different water, different pools, different river, and half of them are very successful. All of the algae, blue-green algae, removed. Very clear water after 1 to 2 days.
So we should congratulate you for developing this.
But not always successful.
Yes, we are very happy.
And the use of the enzymes has a very high environmental risk because the blue-green algae, after the breakdown of its cell membrane, it releases toxic substance. So in one trial, when the owner of the pond, he deposits the enzyme, and for 1 day, the blue algae disappeared. Then he deposits the shrimp for growing, and most of them
They die.
use the toxic substance.
Oh.
I suggest him wait for 3 to 5 days and let all of the toxic substance oxidize by oxygen, but he did not wait.
Okay. So that is one more query. There is not a question.
Then I tried for 18 months to solve this problem. I isolate bacteria from shrimp growing river. from a mud of the river, and I get a bacillus bacteria. It can remove the toxic substance from blue-green algae very efficiently in few hours. So now the enzyme is combined with the bacteria together to be a product. Yes.
I think you can say because another 5 minutes we will have this session. Okay,
I welcome the question.
Yeah, no, there is not question, just discussion because we discuss so many. One more thing is that whether students, like we have some students here, okay, they can be involved in all these short-term projects where they can get training or something like that. We can, with such type of problems your students are getting up there.
Yes.
They have trainings in companies like you are producing enzymes.
Yes.
The students are working. Okay. So are they that paid as a salary or they are paid hourly basis or like that?
Yeah, I paid. But if a freshman, maybe a new master student wants to work in my lab, at least 1 month training, at least. I teach her how to use equipment in my lab, and for the safety. And something about the procedure, for example, the detection of the sugar, the test of enzyme activity, including cellulase, including xylase, lactase, and how to inoculate in microbes, and how to make the substrate in the flask in a fermentation tank. I have to teach them for at least 1 month training. It's very important.
Okay, and then how much? I just want to ask master student.
Okay.
They get fellowship. They
Yeah,
get
fellowship, yes.
Okay, and they also get some if they are working.
Payment, yes.
Yeah, payment also. So it is on hourly basis or it's on monthly basis or weekly basis.
Okay.
Or whatever they work, they do, they get some share of out of it. Very complicated question.
Yeah, very complicated.
Yes. Okay, no need to tell, but they are financially helped. Okay, so it means that we work on all these aspects, and we are having some products.
Sometimes I judge the productivity of students according to this job.
Yeah.
According to the results.
So what is your feedback about our Indian students? You are yeah.
I will take Dr. Puneet Singh as an example.
Yeah, as example. Yeah.
If we can finish what I asked you to do, for example, to establish a new model of a new protein in 1 month, it will be very good.
Okay.
If we fail,
So
it's a big
oh, it's big problem.
I will have a long talk with you.
Yes, it is. It is always. Yes, no, it's good thing actually. If we succeed, it's good. If we fail, big problem. Okay, so it is true everywhere.
Because in my lab, we are not always successful.
In every lab, we are not always successful. Sometimes we get success, sometimes we don't get. Okay, and I think it was a very good interaction today. And if the students they can have another 5-7 minutes are there, if they can have, otherwise we'll try to finish it after 6 minutes. Any questions, any discussion, please? You can have any queries or something. So I think Kaki, you can announce the 5 minutes are left, I think. Huh? So much thing. So I think you should close on that. Once more you can ask if somebody is interested. We have little time left.
So, any more queries you have? I think our professors have really explained very well.
I think today they have got 3 type of
Yeah,
lectures.
we already discussed.
Very exhaustive. So they need to discuss many things. And then I think after this on Saturday we will have exam. Okay. So we will tell you the details. I think we will brief. I think.
So with this, we come to the end of the tutorial section. Now we'll be having a tea, and then we can disperse to our places. Thank you.